Hello everyone, this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati. And what we were discussing about the different aspects of the cell and molecular biology in this particular course. So far what we have discussed about the origin of life and the evolution in the first module. Followed by we have discussed about the cellular structures, followed by we have discussed about the cell growth, apoptosis, and what are the different parameters to monitor the growth and so on. And in this previous module, we have also discussed about the cellular transport and how the cells are taking up the nutrition from the environment and how they are actually going to take up the molecules, what are the different mechanisms in which the molecules are being transported inside the cell, and within the cell how the cell intercellular trafficking system is distributing these material to the different organelles. Now these molecules are interacting with the cell, and as a result these cells are responding to these treatments, and these treatments are mostly being, sometimes they are pro-growth supportive, which means they are supporting the growth of the cell. Sometimes these molecules are not good for the cell, so the cell is responding to in such a way that either it is going to be go for the apoptosis, autophagy, or the necrosis. In either of these cases, the cell is continuously responding to the environment, whether it is the molecules, whether it is the environmental factors such as light, water, pH, humidity, and so on. And these responses comes from a very well developed network of proteins what is present inside the cell. This protein network is taking up the responses from the cell surface, and then it is delineating these responses to the nucleus, and as a response to these treatments, the nucleus is modulating the different types of activities. Like for example, if there will be a cell which is responding to the glucose present in the environment, right, then the cellular system is going to start responding in a positive mode, and it is actually going to start synthesizing the molecules which is required for the metabolism of this particular molecules, right? And all these events are tightly regulated, all these events are interrelated to each other, and all these events are part of the cell signaling, which means where the cell is actually going to respond to the external environment. So in this particular module, we are discussing about the cell signaling and what are different components of the cell signaling, and how these components are connected to each other, how these components are regulating the whole event, and what are the downstream effects when these components are coming together. So as you can see that the cell signaling is a very important parameter, right? So it is required for the different types of functions. So it is required for the cell growth and division, it is required for the cell division, it is required for the metabolism and hemostasis, immune response, cell death, and so on. So what are different functions of the cell signaling? So it regulates the cell growth and the division, which means it is actually going to ensure that there will be a proper tissue development and the repair. So first aspect is that it is actually going to regulate the cell growth and the division. Then it is also going to control the cell differentiation, which means if a cell is differentiating, it is making the different types of organelles or different types of cells, then that also has to be tightly regulated. So the first event is that it is going to control the cell growth and division. Remember, we have discussed a lot about this when we were discussing about the cyclin and cyclin dependent protein kinases and how they are regulating the switching from the G1 to S phase or S to G2 phase and G2 to M phase and so on, right? Apart from that, it also going to regulate the cell differentiation, which means it is going to regulate the stem cells or the primary cells to get go for the differentiation and form the specialized cells. Then it also going to regulate the metabolism and the homeostasis. One of the classical example is what we have discussed when we were discussing about the glucose, right? So when there will be an uptake of glucose, the glucose is going to be utilized into the downstream carbohydrate metabolisms, right? So that is required for maintaining the internal balance by responding to the nutrients and the energy levels, right? And then the number 4, it is actually going to mediate the immune responses. So this is number 1, this is number 2, and this is number 3. And in the 4th aspect, where it is actually going to control the immune responses, so it helps to detect the the pathogens. It is going to help to initiate the inflammations and to coordinate the defense. Then we also required the cell signaling for regulating the cell death, right? So we have discussed about the apoptosis. There are so many caspases, initiator caspase and executor caspase are required. So that also is required for eliminating the damage or the unnecessary tissue in a controlled manner, which means we are talking about the apoptosis. And then we also going to it also going to facilitate the communication between the cells, right? So there are neurotransmitters, there are different types of cells which communicate with each other with the help of the cell signaling. So it enables the coordination in development, repair, and to the immune functions. All these examples we are going to see when we are going to go through with this particular topic in this particular module. And then we also going to integrate the environmental cues. So basically the cell signaling is going to detect different types of environmental factors. So it is going to different about the the abiotic stress. It is going to detect the toxins. It is going to detect the light, osmolarity, pH, and humidity, and all those kind of things. And that is going to help in maintaining the internal environment or the homeostasis. Then it is also going to support the tissue and the organ functions. So it is going to synchronize the activity across the different tissue for the proper organizations. For example, if you have studied the digestion, right? So digestion starts from the stomach, then the food goes into the small intestine, then the food goes from there to the large intestine, and then it is being excreted out. All these compartments what are present within the digestive systems are coordinating with each other, right? Once the digestion in the stomach is over, then only the pyloric sphincter is going to be open, and then only the food from the stomach will enter into the small intestine. And same is the story when it goes from the small intestine into the large intestine, so that the water and the other micronutrients could be absorbed from the undigested food, and that's how it is actually going to form the feces. So cell signaling is a very essential and integrated component of the cell, so that it is actually going to perform the so many different types of functions, and all these functions are required for maintaining the homeostasis of that particular cell, so that it is actually going to grow, and it is actually going to differentiate into the different types of cells. Now talking about why there is a need to do the cell signaling, right? So cell signaling is required to survive and function efficiently. Organism may quickly respond to the sudden changes, and this is very important, right? If you don't have cell signaling, you will not be able to respond to the incoming threats, right? And incoming threats probably could come from the many types, right? From many places, right? It could be from internal, it could be from external. A classical example is how the body reacts to the acute stress, like facing a threat or experiencing the fear. For example, when it is a very dark, or when it is lightning, or when there is a cloud bursting, and all those kind of things. So it actually creates the fear, right? And it creates the fear so that your body should be ready for incoming problems, right? Incoming threats. And this situation activates the different types of highly coordinated signaling events. For one of the classical example is fight or flight response to this stress, right? Suppose you see a lion, right? Or if you encounter a lion, or if you encounter a dog, for example, in your street, right? Now, when you encounter a dog, you have two options. Either you will go and fight with this particular dog, and it will, he will run away, or you will actually decide that, okay, I will not fight, I will run away. Okay, so these kind of decisions comes as soon as you see the particular threats. If the threat is manageable, like if it is a lion, you will decide to run away. If it is a dog, you probably could think about whether I could be able to manage this particular thing or not. If you can manage, then you will fight. If you cannot manage, then you will run away. So these kind of options comes, or these kind of things comes by a response which is called as fight or flight responses, and these are all linked to the cell signaling. So cell signaling, one of the integral portion of the cell signaling is the perception of the stress, right? You have to first detect the incoming threats. You have to detect the incoming stress, right? And that occurs, or that happens within the brain, right? So the brain, especially the hypothalamus, detect a threat and activate the sympathetic nervous system. So it actually signals to the adrenal glands, or it actually systematic hormonal signaling. So it gives the signal to the adrenal glands, and the nerve impulse stimulates the adrenal medulla, which secretes the epinephrine, right? And that epinephrine is actually going to ask for you to decide whether you want to fight or whether you want to flight, because accordingly the body is going to prepare the muscles, body is going to prepare the blood pressure and those kind of events, and that's how it's actually going to control those events. So then adrenal travels to the multiple organs and binding to the adrenogenic receptor onto the target cell, and that's how it is actually going to prepare the body for the different types of threats. And then you are going to have the third event, which is the cellular responses. So if you see the cell signaling is a multi-step process. In the first step, you are going to respond to the stress. In the second event, you are going to ask the cellular system to respond. So this triggers the various intracellular signaling cascade that leads to the increased heart rate and blood pressure to pump more oxygen, dilation of the airway to improve the oxygen intake, right? Conversion of glycogen to glucose, so that you have ample amount of glucose available, so that it can be utilized when you will want to run away and those kind of activities, to provide the immediate energy, right? Because the immediate energy comes from the glucose, not from the fat or other biomolecules. Then it's going to reduce the digestive activity so that you can conserve the energy, and it is also going to dilate the pupils so that you can be able to even look at under the very low light conditions, so that more and more light will go into your eyes. So these are the cellular responses what are going to happen when you are going to encounter a lion or the dog, and under the fight and the flight responses, which is going to be governed by the cell signaling. Then there will be a contraction of the skeletal muscles, which is going to improve the strength and the responsiveness, right? And what will be the outcome? The outcome is the organism becomes physically and mentally prepared for the fight or the flee, right? And these two events are very important, right? Because the last time judgment, whether I could be able to fight to a particular stress or whether I should flee away from this particular place, is what you are going to do, right? So fight and flight response actually is a response where the whole body is involved, right? You are actually going to dilate the eyes so that you can actually be able to see under the dim light. the brain is actually going to give you the signal to the adrenal glands, and adrenal glands are going to start secreting the epinephrine, and that epinephrine is actually going to increase the blood pressure. It is actually going to increase the supply of blood to the heart and other things, and then your heartbeat will go up, so that more and more blood should be supplied to the different organs of the body. And then it is actually going to start catalyzing the the conversion of the glycogen into glucose, so that there should be more amount of glucose what is going to be present into the bloodstream. And then it also going to prepare the muscles, so that muscles should have more amount of oxygen and more amount of glucose, and they will be ready to act on, right? So they will be ready to fight, right? And then there would be a retardation or the inhibition of the digestive system, so that the more and more energy should be utilized for this particular response. Now, as far as the cell signaling is concerned, the cell signaling in the organism could be of three different types. It could be a autocrine signaling, it could be a paracrine signaling, or it could be endocrine signaling. And all these signaling events can be compared with the different types of interactions. For example, when you interact with your friends who's sitting next to on your bench, right? Or you can actually talk to the same friend when he is in home, or you can talk to the same friend when he is in the another city or another country. So the autocrine signaling means when you are going to talk to yourself. Okay, so if you compare it with that, so you talk to yourself. Many times it happens that you talk to yourself when you are making a plan, when you are arranging the material, or when you are, suppose you are going for a trip, right? Then you talk to yourself and say, okay, I need these are the material, these are the cloths I require, these are the dress material I required. So you first make the list, and then you talk to yourself and start taking what are the materials I have taken, what are the materials I have left, and so on. And then we have the paracrine signaling, where you are actually going to talk to your friend who is on your neighbor, or who is in the next bench, which means very close, right? And then the endocrine signaling means you talk to your friend, but he is in the another city. Okay, so first we'll discuss about the autocrine signaling. So autocrine signaling means when you are going to talk to yourself, which means a cell is there, this is a nucleus, and the cell itself is going to secrete a material, right? Or still start secreting a ligand, and that ligand will go and bind to a particular receptor onto the cell surface, and then it is going to start sending the signal to the cell, right? This happens because you want a very good microenvironment around you. So every cell is secreting the cells, the molecules outside, so that it is actually going to maintain a local environment, and that molecules are actually going and binding to the receptor and again returning back to the cell, right? So this actually makes the microenvironment of a particular cell, and that gives the pro-supportive growth to this particular cell, and that's how this cell is feeling good actually. So what is autocrine signaling? Autocrine signaling is a form of cell communication in which a cell secretes signaling molecules such as cytokines or the growth factor, and that binds to the receptor on the same cell that produce them. In some cases, the neighboring cells of the same type can also respond to these signals. This type of signaling allows the cell to regulate its own activity and reinforce the specific responses. It is especially important in cell growth and differentiation, immune cell activations, inflammatory responses, and the embryonic development. Cell growth and differentiation we have discussed a lot when we were talking about the the transition of the cells from G1 to G2, S phase and S to G2 phase and all that. Even for the differentiation also, the many type of cells are secreting the molecules, and then they are coming back and binding to a specific receptor, and that's how they are generating the specific type of cell signaling. And that cell signaling is also changing the transcriptomic analysis or transcriptome of a particular cell, or in eventually the proteome of the cell, and basically changing the outcome, right? For example, if a cell wants to change into the epithelial cell, right? If the stem cell wants to change into the epithelial cell, it is actually going to secrete some of the signaling molecules so that it will bind to its own receptor, and then that receptor downstream signaling is going to start changing the different types of transcription factor are present within the cell, and as a result of that, it's actually going to start activating the transcription of the specific genes, and the gene product of those genes or gene products are actually going to start changing the outcomes. Similarly, you have the immune cell activations that anyway we are going to discuss in detail in a future module, but you have seen the T cell, how the T cell is getting activated, how the B cell is getting activated. So it is secreting the antibodies, and these antibodies are binding to the B cell on its own, and that's how it is actually increasing the proliferation of B cell, and that's how a particular clone, when it is being selected for converting it into the plasma cells so that it's actually going to start secreting the antibodies, is being done by this way only, right? So you know that when there will be a clonal selection, the antibodies are going to be secreted, and then the receptors are going to be engaged with a particular antigen or those kind of thing, and that's how it is actually going to be remain into the proliferation stage, and that's how the clonal selection works. In fact, for other cell type also, or other immune cell type also, the autocrine signaling is activating and asking them to secrete the different types of cytokines. Then we also have the inflammatory responses. The same way, the inflammatory cells like mast cells and some of the dendritic cells and those kind of cells are also secreting a lot of inflammatory molecules when they are going through the autocrine signaling. And then we also have the similar way, the embryonic development and so on. So there are cell signaling events controlling this in a very regulated manner. So autocrine signaling ensures the rapid localized feedback, and it is critical in tightly controlled processes like T cell proliferations, cell fate determination using the development. So, and then you also have the pain perception and the apoptosis during infections. Now let's take the examples. So we have an example of the T cell activation. So this is the antigen presenting cells. This is the naive T cells, and when the antigen presenting cell is presenting the antigen onto the MSCs, it is being recognized by the T cell receptor present on the T cells, and that's how it is actually secreting the IL-2, and then these IL-2 are binding onto the T cell, and that's how it is converting this naive T cell into activated T cells. So autocrine signaling play a crucial role in the activation and clonal expansion of the T cell, particularly the CD4 positive T helper cells during an immune response. So antigen recognition, so a naive CD4 positive C cell recognizes a specific antigen presented onto the MSC class 2 via its T cell receptor, and then these active interactions along with the co-stimulatory molecule activate the T cells. And then there are so many, the cytokines are going to be secreted from the naive T cells. So there will be an IL-2 secretion, and there will be a receptor expansions. So the activated T cell begin to produce IL-2 and simultaneously upregulate the IL-2 receptor on its cell surface. And then IL-2 binds to the IL-2 receptor on the same T cell and forming an autocrine loops, which means these IL-2, when they will bind to the IL-2 containing T cells, they will go and bind, and then they will ask for secretion of more and more IL-2, and that's how there will be the expansion of this activation. And then this signaling drives the proliferation of the activated T cells, creating more identical effector T cell capable of responding to the antigen. So, and then there is another example of regulation of the apoptosis. So IL-2 signaling also influence whether the activated C cell will survive or undergo the programmed cell death, helping the immune balance. This autogrind loop ensures that the only antigen specific T cell expand, making the immune response both efficient and highly specific, right? So this is just a single example how the autocrine signaling is helping the immune responses and how it is helping the T cell activation and as well as the differentiation. Now let's move on to the next mode, and the next mode is called as the paracrine signaling. So as I said, paracrine signaling means talking to your friend who is sitting on the next bench or sitting in his home, right? So paracrine signaling means this is you, and this is your friend. Okay, so you are talking to your friend, which is not very close, but it is not very far also. So you can be, so what you are doing is you are secreting the molecule. It means you are sending the signal, and your friend who is actually having the receptor for this particular signal is receiving those signals, and that's how this cell is actually going to respond to your signal. Okay, this means you are actually going to talk to your friend, and he is actually sitting next to you. So this is only if it is adjacent target cells, right? So in paracrine signaling, the signaling molecules can be secreted in the extracellular fluid. These molecules act as a local mediator and target the cell neighboring the signaling by the local diffusion. The signaling can aid in exchange of information between the cell using a soluble signal that can diffuse one cell to another cell, which means this is a signaling which is happening within the short distances. The signaling can have high local concentration of the signaling molecules. The signal can include the growth factors, cytokines, hormones, prostaglandin, etc. So this is the signaling molecules, right? So it is secreting the molecules, and then it is being received by the target cell which are present within the vicinity. So it is secreting the molecule which is being received by a target cell which is within the vicinity, and that's how it is actually amplifying this signal. So one of the classical example is neurotransmitters carry signal between the nerve cells using the paracrine signaling. Upon being released at the postsynaptic end, the neurotransmitters diffuse across the synaptic gap to an adjacent postsynaptic neuron or the effector cells. Another examples can be of the action of cytokines on the neighboring cell using the immune responses and the inflammation. Testosterone released by the leading cells, right, bind a receptor, androgen receptor, in neighboring Sertoli cells to regulate the spermatogenesis. And then you also have an example of cell differentiation, proliferation and migration during the embryonic developments are regulated by the paracrine signaling by the factors such as growth factors. So paracrine signaling means talking to your friend sitting adjacent to you. So you're giving a signal maybe within the your classroom, right? So for example, if you're trying to give some signal to your friend who's sitting in the same room, that also within the paracrine signaling. Then we are coming back to the third one, and that is called as the endocrine signaling. So endocrine signaling means talking to your friend who is sitting in a next city, okay, or very far away. So what you're doing, the signaling cell is secreting the ligands or signaling molecules. Then it is going through the bloodstream and then reaching to the target cell which is very far away. For example, you are secreting the molecules from the brain. For example, you are secreting the molecule from the brain, and then it is reaching to liver. That is the endocrine signaling. So endocrine signaling involves the release of signaling molecules called hormones by a specialized endocrine cells. these hormones travel throughout the bloodstream to act on a distant target cells, right? So these are the cells, these are the target, these are the secretory cells, right? They have secreted a molecule which will into the bloodstream, and then it will reach to a target cell, and this target cell is actually going to have a receptor, and that's how it is actually going to bind. The endocrine cells are typically located in the endocrine glands such as thyroid, pituitary, and the adrenal glands, and they will secrete the molecule, and then it will reach to the target cells. Many examples, we have many examples. So endocrine signaling can happen in different types of organisms, right? So it is mostly being done by the hormones, right? So, and hormones could be present in all the eukaryotic cell, right? So mostly the hormones are present in the multicellular organisms, right? So hormones are being present in the animals, right? Or hormones could be present in plant, and they are regulating the different types of activities, right? So they are regulating the metabolisms, they are regulating the cellular functions, and so on, right? So in the endocrine signaling, first we are going to discuss about the animal hormones, and the second we are going to discuss about the plant hormones. So the first is animal hormones. The term hormone was first used by the physiologist Ernest Henry Starling in 1905. Hormones were either described as a substance produced by the gland with internal secretion which serves to carry signal to the blood to the target organs, but these were many exceptions. Thus, there have been some way to define them. One such definition is a hormone is an organic compound secreted by the plant or the animal that helps to regulate the physiological processes and maintain the internal balance or the homeostasis. Now first talk about the animal hormones. So animal hormones are widely distributed throughout the body, right? And they are doing different types of functions. Animal hormones are regulating the growth and development. Animal hormones are regulating the metabolism and energy. Animal hormones are regulating the reproductive system. Animal hormones are regulating the flight and fright responses. They are regulating to the different types of stress like injury, stress, and environmental factors. They are also maintaining the mood and the behavior, and then also maintaining the homeostasis, and they are also maintaining the sleep or the sleep cycle. So hormones are the chemical messenger that transmit the signal into the bloodstream and the tissue. They act slowly and influence a wide variety of functions. Imbalance in hormone produced can lead to the various disorders. One hormone can regulate more than one biological functions. Multiple hormone can also be involved with the regulation of one biological functions, and the hormone production can be regulated by a negative feedback controls, right? Chemically, hormone can be classified into the two main groups based on their solubility. So you can have the lipid soluble hormones or the water soluble hormones. So what is the general mechanism of hormone signaling? So the endocrine glands release the hormones into the extracellular space, from where they enter into the capillaries and travel throughout the bloodstream to reach the distant target cells, enabling the systematic effect. So this is the endocrine gland, right? And it is actually secreting the hormone. Then it goes into the bloodstream. Then hormone producing the cells are specialized and release the hormone via the exocytosis or the other membrane transport mechanisms. These transport mechanisms we have discussed in detail in the previous module, right? How the molecules are being transported, right? And then they reach to the target cell or their target organs actually. So this is the endocrine glands, right, which is secreting the molecules into the bloodstream. Then it travels throughout the bloodstream, then it reaches to the target cell, and then the target cells are actually having the receptors, right? And then these receptors are binding these hormones, and that's how they are actually regulating or relaying the signal within the cell. Sometimes these receptors are not present outside. These receptors might be present inside the nucleus, and accordingly the animal hormones could be of different types, right? As we said, animal hormones could be lipid soluble hormones or the water soluble hormones. Target cells for a hormone can be of various cell types distributed across different tissues. Hormones act by binding to a specific receptors either on the cell surface or within the cell depending upon their chemical nature. This receptor binding initiates the downstream signaling events which may ultimately lead to the change in the gene expressions. The response of target cell may be detected by the original hormone producing cells, resulting in a reduced hormone control. This response is a negative feedback mechanism, which means if this is the hormone producing cell, right, or hormone producing organ or the glands, when it is secreting the hormone, right, and hormone is reaching to a receptor, right, containing cell, right, so and then when it is binding to this cell is also started responding, and then this cell is actually going to secrete the molecules, or it is actually going to give you the signal to these glands that okay, there we have enough amount of hormones and there is no need to secrete any more hormones, and that's how this hormone production is also going to be reduced or it is going to be lowered down, right? And this kind of mechanism is called as feedback mechanism. Feedback mechanism means I have enough amount of supply, stop supply by enemy. Okay, so that's how this is called as feedback mechanism, which means where you're going to give the feedback to your supplier that okay, I have enough quantity of this particular material, there's no need to secrete or they need to supply it anymore, and that's how it is actually going to reduce the supply. Now, based on the chemical nature, the hormones could be of three different types. It could be a peptide hormones, it could be a steroid hormones, it could be a monoamines. So let's talk about the peptide hormones. The peptide hormones are composed of amino acid linked by a peptide bond, which length ranging from 3 to 200 residues. Sometimes may also be glycosylated, affecting their stability and activity. They are water soluble and cannot cross the plasma membrane. It means they will actually going to bind the receptors what are present onto the cell surface. Therefore, they primarily bind to the cell surface receptor to initiate the cell signaling. Although rare, some peptide hormone can also act through the intracrine signaling, where they function via the intracellular or nuclear receptor within the same cell that synthesizes them. These hormones vary significantly in terms of size, structures and biological functions, and regulating the processes such as metabolism, growth, fluid balance and the immune responses. Peptide hormone producing occurs in a specialized secretory cells rich in endoplasmic reticulums and the Golgi apparatus, which are responsible for the protein synthesis, modification and packaging. Remember when we were discussing about the intracellular trafficking when we were discussing about the cellular structures, you have seen that the endoplasmic reticulum, Golgi bodies and lysosomes, these are the three organelles which are very active in terms of the regulating the cell secretions, right? So the cells which are actually going to produce the hormones are actually going to have a very high quantity of these organelles. Hormones are stored in a secretory vesicles which released upon the appropriate stimulus, allow for a rapid response to the physical cues. The main step in the synthesis of, so how the synthesis of the peptide hormone works, right? So main step in the synthesis of peptide hormone includes first the transcription, the second is post-transcriptional modification. So in the first step, the DNA is going to form the messenger RNA, that is called as transcription, right? And all these events we are going to discuss. How the transcription works, how replication works in the subsequent modules. And then the messenger RNA is going to be crude messenger RNA, right? And then it is actually going to be, do the post-transcriptional modifications, and that also we are going to discuss in a subsequent module. Then the messenger RNA is going to undergo the capping, splicing and polyadenylation to form the mature messenger RNA. And then once it forms the mature messenger RNA, then the mature messenger RNA is going to produce the peptide hormone, and then peptide hormone is going to put into the secretory pathway, and that's how it is going to be secreted out of the cell. So then it is going to be, once the mature messenger RNA is going to be formed, then ribosome on the rough RER translate the messenger RNA into a precursor polypeptides. And then co-translational and post-transcriptional modification signal peptides are cleaved to form the pro-hormones, and it may be folded, glycosylated and cleaved into the active form before being packed into the vesicle for storage and release upon stimulus. These peptide hormones can be of different types depending upon the type of organ it is actually going to produce. So the peptide hormones can be classified based on their site of synthesis, as this determine their specific functions, target organs and the physiological roles. Now let's see about the different types of peptide hormone. And as I said, peptide hormones are being classified based on their origin, right? So peptide hormones could be secreted from the hypothalamus. So remember that a peptide hormone is going to be secreted from the different organs. It is going to be secreted from the digestive system, it is going to be secreted from the brain, it is going to be secreted from the other endocrine glands. So the first is hypothalamus, right? So from the hypothalamus, a large number of peptide bonds are going to be secreted. One is antidiuretic hormone or ADH, right? And ADH, what is the function of the ADH? It is actually going to promote the water reabsorption in the kidney and regulate the blood osmolarity. And if there will be a deficiency of this particular hormone, the person is going to develop the diabetes insipidus, where you are going to have the excessive urinations and the dehydrations. Then we have the CRH or the chondrotropin releasing hormone. It actually stimulates the ACTH release from the anterior pituitary, and if there will be not enough quantity of the CRH, then there will be a lower production of the acetylcholine receptor or ACTH, and that is actually going to be responsible for the adrenal insufficiency or the fatigue. Then we also have the growth hormone releasing hormones. So that also is going to stimulate the secretion of the growth hormone from the anterior pituitary, and the growth, if there will be a lower production of the growth hormone, then there will be a growth retardations, especially into the children. So that will actually going to cause the dwarfism, right? And then you also have the gonadotropin releasing hormones. So that also is going to stimulate the production of the luteinizing hormones and the follicular stimulating hormones from the anterior pituitary, and that also is going to be caused. So if there will be a reduction in GnRH, then it is actually going to cause the hypogonadism and the infertility, because if you don't produce the LH or the FSH, there will be a less production of eggs, and then that is also going to cause the infertility. Then we also have the oxytocin, and oxytocin stimulates the uterine contractions and the milk ejections into the lactating females, and there will be a deficiency of the oxytocin, then it is actually going to cause the poor labor and impaired milk productions. Then we have the somatostatin. The somatostatin is going to inhibit the growth hormone and the TSH secretions, and the excessive can lead to the growth or the metabolism related issue. So don't worry, it's not like that the hormone deficiency only causes the disease. There will be an excess of hormone also can cause a disease. Then we have the thyrotropin releasing hormone or TRH, and that stimulates the thyroid stimulating hormones and mildly prolactin secretion from the anterior pituitary. And if there will be a deficiency of TRH, it is actually going to reduce the production of TSH, and that is actually going to cause the hypothyroidism. Then we also have the peptide hormones from the anterior pituitary. So this is another part of the brain from where you are going to have the peptide hormones. So you have the ACTH, and ACTH is stimulating the cortisol secretion from the adrenal cortica, and if there will be a deficiency of ACTH, it is actually going to cause the fatigue or the low blood pressures. If then you have FSH, or the follicle stimulating hormones, that stimulate the follicle development in the ovary and spermatogenesis in the testes. If there will be a deficiency of SH, then it is going to cause the infertility, irregular menstrual cycles, or the reduced sperm counts. Then we have the LH, or the luteinizing hormones. So this LH is going to trigger the ovulation and the corpus luteum formation in the females and the testosterone production into the males. And if there will be a reduction in the LH production, then it is going to cause the infertility, menstrual disorders, and the low testosterone. Then we have a growth hormones, and growth hormone stimulates the growth and cell reproduction and regenerations, and there will be a reduction. Then it is going to cause the stunted growth into the children's, fatigue, and the low muscle mass into the adults. Then we have a prolactin. The prolactin stimulates the milk production into the lactating woman and inadequate lactations. Then we have a TSH, or the thyroid stimulating hormones, which stimulate the thyroid hormones productions, and it causes the hypothyroidism, weight gains, or the fatigue. Then we have the peptide hormones, which is from the parathyroid glands. So parathyroid glands are releasing the calcitonin and the parathyroid hormones. Calcitonin is lowering the blood calcium level by inhibiting the osteoclast activity and promoting the calcium excretion by the kidney. And if there will be a reduction of calcitonin, then it is going to cause the mild effects in humans, deficiency usually not clinically significant. Then we have a parathyroid hormone, or the PTH, that increases the blood calcium by stimulating the osteoclasts, enhancing the calcium reabsorption into the kidney and activating the vitamin D. And if there will be a reduction of PTH, then it is going to cause the hypocalcemia, muscle cramps, and tetany or the seizures. Then we also have the peptide hormones from the heart, right? And it is going to be atrial natriuretic factor peptide, or ANP. And ANP promotes the sodium and the water excretion by the kidney and lower blood volume and pressure. And if there will be a reduction of ANP, then it is actually going to cause the hypertension and the fluid retentions. Then we also have the brain natriuretic peptide, or BNP, and that is similar to ANP but secreted mainly from the vertical under pressure or the volume overload. This reduces the ability to compensate to the cardiac stress and used as a heart failure markers. Then we have the peptide hormones from the digestive system. So you have the cholecystokinin, or CCK, and that stimulates the bile release from the gallbladder and the pancreatic enzyme secretions and slows the gastric emptiness. And if there will be a deficiency, then it is actually going to affect the fat digestions, and it is actually going to reduce the satisfaction, which means it is actually going to reduce your satisfaction level when you take the food. Then you also have the gastrin. Gastrin is actually going to stimulate the gastric acid secretion from the parietal cells, and if there will be a deficiency of gastrin, then it is actually going to form the low stomach acid, which means it is actually going to reduce the production of HCl, and that also going to affect the digestion. Then we have the so it is actually going to stimulate the appetite and promote the GH release, and if there will be a reduction, then there will be reduced hungers, possible growth issues in the. Then we have the glucagon-like peptides, and these enhance the insulin secretions, inhibit the glucagon release, and slow gastric emptiness. If there will be a reduction of this or deficiency, then there will be an impaired glucose regulations and possible post-parietal hypoglycemia. Then we have the glucose-dependent insulinotropic peptides, and that stimulates the insulin secretion in response to the oral glucose. And if there will be a deficiency, then it is actually going to decrease the insulin responses to the meals. Then we also have the oxynomodulin, or OXM, right? And that reduces the appetite, inhibit the gastric motility and the acid secretions. And if there will be deficiency, it is going to cause the potential weight regulation imbalance. Then we have a secretin, that stimulates the bicarbonate secretion from the pancreas, inhibits the gastric acid secretions. If there will be a deficiency, then it's going to impair the pH buffering of the chyme, and it is affecting digestion in the small intestine. Then we have a peptide hormones from the liver also. So liver is secreting one peptide hormone, that is the insulin-like growth factor 1, and that mediates many of the growth hormones effects, promotes the systematic body growth, and especially of bones and muscles. If there will be a reduction in this, then there will be a growth failures into children's, reduced muscle mass and the bone density. Then we have the peptide hormones from the kidney. So it's going to be erythropoietin and renin. Erythropoietin is stimulating the red cell production in the bone marrow, and if there will be a reduction of the erythropoietin, then it is actually going to cause the anemia, especially in the chronic kidney diseases. Then we have a renin. So renin is initiating the renin-angiotensin-aldosterone system, and it regulates the blood pressure and the fluid balance. And if there will be a reduction in the renin production, then it is going to cause the hypotensions, electrolyte imbalance, and impaired RAS responses. Then we also have the peptide hormones from the fat tissues or the adipose tissues. So that is called as adiponectin, leptin, and resistin. And the adiponectin is enhancing the insulin sensitivity, inflammatory response, promotes the lipid oxidations. If there will be a reduction in the adiponectin, then it is actually going to cause insulin resistance, increased risk of metabolic syndromes, and the obesity. Then we have the leptin. Leptin regulates the appetite and the energy expenditure by acting on the hypothalamus, and there will be reduction, then it is going to cause the uncontrolled appetite, obesity, metabolic dysregulations. Then we have resistance. Resistance impairs the insulin sensitivity, and it has a potential role in the insulin resistance and inflammation if dysregulated. Then we also have the peptide hormones from the pancreas. So peptide hormones are like insulin, glucagon, amylin, pancreatic polypeptide, somatostatin, and vasoactive intestinal peptides. So insulin is lowering the blood glucose level, glucagon is raising the blood glucose level, and amylin is slowing the gastric emptiness and suppresses the glucagon and promoting the satiety. Then we have pancreatic polypeptide that regulate the exocrine pancreatic secretion and influences the hepatic glycogen levels and the GI motility. Then we have somatostatin. Somatostatin inhibits the secretion of insulin, glucagon, GH, and many GI hormones. And then we have a vasoactive intestinal peptide that relaxes the GI smooth muscles, stimulating the water and the electrolyte secretion into the intestine. And as far as the deficiency is concerned, with the insulin deficiency, it is actually going to cause the diabetes mellitus. With the deficiency of the glucagon, it is actually going to cause the hypoglycemia, or the and it happens especially during the fasting. Then the amylin is causing the postprandial glucose spikes or the impaired satiety. Then we have pancreatic polypeptide, which causes the altered digestive enzyme release and the GI motility. Then we have somatostatin that causes the excess glucose release and rarely isolated deficiency. Then we have a vasoactive intestinal peptide that causes the malabsorptions, and if excess, it causes the deficiency effects unclear. Okay, so these are the peptide hormones what has been secreted from the different organs present within the animal body. Okay, all these peptide hormones are mostly being acting through a specific pathways, right? So their receptors are different, their machinery is different, but their mechanism, how they actually functions, are more or less same, right? So to explain these mechanisms, I have taken an example of glucagon, how the cell signaling of glucagon works, right? So glucagon signaling is different from the insulin signaling, glucagon signaling is different from amylin, but the events are same. When we explain the glucagon, doesn't mean that the other hormones are also going to follow the similar kind of machinery. The machinery is going to be different, but the events are going to be same. Now, talking about the glucagon, it is a very simple and well characterized peptide hormone that play a central role in maintaining the blood glucose level during fasting. How the glucagon is being synthesized? So glucagon is synthesized by the alpha cell of the pancreatic island of Langerhans. It is initially produced as a larger precursor pre-proglucagon encoded by the GCG gene. Inside the rough endoplasmic reticulum, the signal peptide is cleaved to form the proglucagon, which is further processed in the secretory vesicle to yield the 29 amino acid long glucagon. These vesicles store the glucagon until secretion is triggered by a drop into the blood glucose level. So glucagon and insulin are working in a reverse direction, right? So if you have a glucose, right, and if the glucose level will go up, right, then it is actually going to induce the secretion of insulin. If the glucose level will go down, then it is actually going to induce the production of glucagon. Okay, so glucagon's job is to maintain the blood glucose level. Insulin's job is to increase the glucose level. Insulin's job is to reduce the glucose level. So insulin is actually going to take the glucose from the blood and put it into the liver for storage. Glucagon is going to do the reverse. It is going to take the glycogen from the liver, convert that into the glucose and put the glucose into the blood streams. What is the signaling mechanism of the glucagon? So glucagon exerts its effect by binding to the glucagon receptor or GPCR, right, predominantly expressed in the liver and to a lesser extent into the kidney, pancreas and the other tissues. Upon binding, the receptor activates the GS and GQ protein. So this is the GCG receptor. So when the glucagon comes and binds to this, it actually activates the GS and the GQ proteins. GS stimulates the adenylyl cyclase, increasing the cyclic AMP level and activating the protein kinase A, which then phosphorylates the transcription factor like CRB to promote the gene expressions. So when there will be the activation of GS, then it is actually going to convert the ATP into the cyclic AMP with the help of the adenylyl cyclase. And then the AMP is actually going to activate the protein kinase A, and protein kinase A is going to activate the CREB. Okay, and CREB is actually going to work as the transcription factor. So GQ activation leads to the phospholipase C activations. So on the other hand, the GQ is going to activate the phospholipase C, right? And the phospholipase C is going to convert the PIP2 into IP3, right? And which play a role in calcium release from the ER, further enhancing the CREB dependent transcription via the CREB regulated transcription factors. Beside the CREB and CRT-C2 pathway, glucagon is also known to activate several other intracellular signaling cascades. So basically, this receptor GCG is actually having the GS and GQ proteins. When they get activated, the GS is going to activate the adenylyl cyclase, which is going to convert the ATP into the cyclic AMP. Cyclic AMP will go and bind to the protein kinase A, and the protein kinase A is going to activate the CRB. Similarly, GQ is going to activate the phospholipase C, and the phospholipase C is going to start converting the PIP2 into IP3, and IP3 is going to activate the endoplasmic reticulum. So this is the ER to release the calcium, and once the intracellular calcium of the cell will go up, it is actually going to delineate the calcium dependent signaling within the cell, and that's how it is actually going to activate the CRTCS, C2 signaling, and these two pathways are actually going to increase the blood calcium level. These coordinated signaling events demonstrate how peptide hormones like glucagon use multiple intracellular pathways to regulate gene expression and maintain the physiological balance, highlighting the complexity and the precision of the cell signaling mechanisms. Now, how the glucagon signaling is going to be regulated? So, glucagon secretion is stimulated by the hypoglycemia, which means low blood glucose, amino acid intake, and the catecholamine during stress. It is inhibited by the insulin, which signals high blood glucose, and by the somatostatin, which act as a general inhibitor of the pancreatic hormone release. So basically, the glucagon signaling is regulated first at the level of ligands, like first at the level of the factors such as low blood glucose. So if there will be low blood glucose, it is going to stimulate. If there will be high blood glucose, it is going to down regulate the production of glucagon. In addition to that, the insulin, when there will be secretion of insulin, insulin is going to inhibit the production of glucagon. Similarly, the somatostatin is also going to inhibit the insulin productions. Intracellularly, the signal is terminated by the phosphodiesterase, which degrade the cyclic AMP. Remember, in the first step, adenine cyclase is processing the ATP and producing the cyclic AMP, which is activating the protein kinase A. So, if in the cell, the phosphodiesterase is degrading the cyclic AMP, so that there will be no activation of protein kinase A, and that's how it is actually going to seize, or it is going to inhibit the cell signaling. And through receptor desensitization, if glucagon level remains elevated for too long, this tight regulation ensures the blood glucose remain within a narrow physiological range. Now, what is the outcome of the glucagon signaling? So it is actually going to increase the blood glucose level. It is going to stimulate the glucogenolysis in the liver. It is going to enhance the glucogenesis. It's going to inhibit the blood glucose synthesis, and it's going to promote the lipolysis in the adipose tissue, which means it is going to increase the availability of fat for the energy, and also going to maintain the blood glucose during fasting or in between the meals. So this is all about what we have discussed for the animal hormones. So far what we have discussed about the different types of cell signaling. We have discussed about the autocrine signaling, paracrine signaling, and endocrine signaling. Within the endocrine signaling, so far what we have discussed about the peptide hormones. And within the peptide hormones, we have discussed about the peptide hormones secreted from the liver, heart, pancreas, brain, and so on. So with this, I would like to conclude my lecture here. In our subsequent lecture, we are going to discuss some more animal hormones, and then we are also going to discuss about the plant hormone. Thank you.